Hello everyone. In today's lecture, we are going to study the control and prevention of parasites. Controlling parasites is extremely important because parasites reduce animal health, decrease productivity, and can also spread diseases to humans. Parasite control is mainly divided into three major methods: biological control, chemical control, and physical control. Let us begin with biological control. One important biological method involves entomopathogenic fungi. These fungi live in soil and infect insects by penetrating their cuticle. They enter the insect body, kill the insect, and finally feed on it. Another biological method uses nematopathogenic fungi. These fungi infect and kill nematodes or parasitic worms. Certain birds and domestic fowls are also used in biological control because they naturally help reduce parasite populations. Now let us move toward chemical control. Chemical control uses antiparasitic drugs and insecticides. The first important group is benzimidazoles. These drugs are mainly used against nematode and trematode infections. However, they have limited activity against cestodes. The second group is macrocyclic lactones. Drugs like Ivermectin belong to this group and are widely used in the treatment of heartworm disease. Next are imidazothiazoles. These drugs are commonly used in cattle, sheep, pigs, goats, and poultry to treat nematode infections. However, they are not effective against flukes and tapeworms. Insecticides are also widely used to control insect parasites and disease vectors. Now let us discuss physical control methods. Physical control includes the use of light traps, electromagnetic radiation, and good management practices to reduce parasite populations. Now let us understand how parasitic diseases can be prevented. The first preventive method is prophylaxis. Prophylaxis means the use of drugs or preventive measures before disease develops. The second method is good management. This includes biosecurity, hygienic surroundings, and separate grazing systems to reduce parasite transmission. Another important method is immunological control. This includes vaccination and breeding animals that are naturally resistant to parasitic infections. Lastly, the refugia method is used to slow down the development of drug resistance in parasites. This method helps maintain a population of parasites that are still sensitive to treatment drugs. In the next lecture, we will discuss the branches of parasitology, vectors and their types, and other important common definitions related to parasitology. Thanks for watching. Don't forget to like, subscribe, and keep learning. See you in the next lecture.